Hello, everyone, and welcome to this audio podcast. This audio podcast, as part of a Phantom edition. This audio podcast, as part of a Vertex Roulet. This audio podcast is part of a Lobostics Machina. This audio podcast, as part of a Storm Lobostics production. In through this audio podcast, in today's audio podcast, a look at works from 2022, works as a research poster presentation put forward for the International Pediatric Transplantation Association, or IPTA 2022, and this research poster presentation looks at transplant neuroimmunobiology, the immune system, transplant rejection, and neuroendocrinology, a retrospective. This research poster presentation was collaborative between between three three academic institutions, in between three academic institutions. And so to begin with some aims, looking at some aims. So the aims of this research poster presentation of this works were to highlight how specific immunobiological and neuroendocrine markers can reduce chemokine receptor sort of migration, and a receptor migration in across where acute allograft rejection, or AAR, is evidenced. In around the mechanisms or the mechanistics, the pre looking at in line to evidence on the literature. The pre-transplant phase is just as central as the post-transplant duration, and it is postulated that the Hansen's general adaptation syndrome, the Hansen's general adaptation syndrome from 1936, and specific neuroimmunobiology and neuroendocrinology should be considered aligned to transplant immunobiology in context of allograft prognoses. In regard to some background works, or background literature. So cytokines are small molecules that signal between cells, inducing growth differentiation, chemotaxis activation, enhanced cytotoxicity, and/or regulation of immunity. Chemokines are cytokines which direct cell migration and/or activate cells. Chemokines are closely related to cytokines, which are produced by many cell types in response to infection or physical damage. They activate cytokines activate and direct effector cells to sites of tissue damage, and regulate leukocyte migration or trafficking into tissues. In context of immunobiology, CC chemokines are chemotactic for monocytes. CXC chemokines are chemotactic for polymorphonuclear morphonuclear neutrophils, or PMNs. Chemokine receptors are expressed on specific cell populations, permitting different chemokines to have selective immunobiological activity. Chemokines are primarily involved in attraction of leukocytes. Leukocytes also, where leukocytes include lymphocytes, monocytes, and neutrophils, and they can be released by many different cell types and serve to guide cells involved in innate immunity, and lymphocytes in adaptive immunity. Some chemokines also have roles in the have roles in the development of lymphocytes, migration, and angiogenesis, where angiogenesis is where in regards to the growth of new blood vessels. It is evident that cytokine molecules are potent molecules for activating and directing effector cell populations to the site of damage, as well as for controlling leukocyte migration into tissues. To attain long-term transplant prognoses or survival post-transplantation has been to attain long-term transplant prognoses post-transplantation, there has been and has been a sort of an ultimate sort of challenge for a variety of solid organ specialties. Immunosuppression, or IST, for example, reduces activation or efficacy of the immune system. Some portions of the immune system itself have immunosuppressive effects on other parts of the immune system, but clinically induced IST is carried out through pharmacotherapy. This is usually done to prevent the body from rejecting an organ transplant for the treatment of autoimmune diseases. When an organ is transplanted, the immune system of the recipient will recognize it as a foreign tissue and attack it. Cortisone the first was the first immunosuppressant, or IST, identified. The more effective azathioprine was identified in around the 19 in around just before the 1960s, but it was not until the discovery of cyclosporin A, or CSA, in around the 1970s that transplant surgery found a sufficiently powerful IST. Monoclonal antibodies, or MABs, are antibodies that are identical to the immune system because they are produced by one type of immune cell. All clones of a single parent cell. Monoclonal antibodies have advanced from involving fusion of immortal cell, which is where an immortal cell, which is in regard to a myeloma tumor cell with a predetermined antibody producing B cell. This fusion is done by ensuring cell membranes are more permeable. Initially, mouse monoclonal antibodies did not make ideal pharmacotherapy or therapeutics because of differences between human and murine genetics. This has been dealt by humanizing murine antibodies, or by making fully human monoclonal antibodies. It has now become standard, for example, to use monoclonal antibodies with IST. Now looking at a particular question as part of this research poster presentation from 2022, looking at how does neuroimmunobiology and neuroendocrinology associate. So stress is a medical terminology for a broad range of strong external stimuli, both physiological and psychological, which can cause physiological response called the general adaptation syndrome, first described in 1936 by Hans Selye in the journal Nature. Selye's works described the general adaptation syndrome as having three stages. One, alarm reaction where the body detects an external stimuli 2, adaptation, where the body interacts defensive countermeasures against the stressor, and 3, exhaustion, where physiologically the body runs out of defenses. It would seem acceptable that stage two, i.e., adaptation, where the body interacts in defensive countermeasures against a stressor, that that it would seem acceptable that stage 2 corresponds to episodes of acute allograft rejection, or AAR. Investigations of the immune system and psychology have become a direction which is significant and clinically important. A change in the amount of natural killer cells, for example, and stress influence activity is an important defense mechanism of the body. It constitutes a component of the preparation for the defense against potential pathogen penetration. When individuals, or when people, for example, get angry, or when there is a sense of anger, the entire body responds. It instantly becomes tense, and physiologically, automatically the body moves towards a fight or flight. Um, um, in a fight or flight. And just to inform here, listeners, that the fight or flight sort of mechanistics, and there is literature around the fight or flight mechanisms or mechanistics. The fight or flight was the fight or flight sort of and the fight or flight context was initially described by Walter Cannon, Walter Cannon in around 1924. Um, in around 1924. So when relaxed, then physiologically the entire becomes relaxed. So stress hormones can change immune cell behavior and the activity of the entire immune system. Studies involving transplant immunobiology and neuroimmunobiology should be included in investigations to better understand acute allograft rejection, or AAR. Now looking at, as part of this research poster presentation, the some of the underlying literature from a retrospective basis. So works by O'Leary in 1990 highlight that chronic stress has been associated with the suppression of immune function, and there is evidence that the immune system may not adapt over time. Works by Adder and Cohen in 1993 highlight both conditioning and stressful stimulation exert biologically effects and stressful stimulation exert biological effects that can alter development or progression of immunologically mediated pathophysiological processes. Works by Dabar and team in 1995 informed that significant and selective changes in numbers and percentages are identified in peripheral blood leukocyte during stress in subpopulations of laboratory rat models. In works by Evans and team in 1995, who examined stressful events and in around anxiety, where the team, when investigators had identified that stressful events and anxiety may affect cellular immunological parameters in subjects with and without human immunodeficiency virus or HIV infection, and cytotoxic T lymphocyte phenotypes. In works by Mills and team in 1996, who looked at and investigated the associations and relationships between psychological characteristics and immune responses to an acute laboratory stressor, and such distribution of in particular cells may significantly affect the ability of the immune system to respond to potential for the immune system to respond to potential or ongoing immune challenges. In works by Shedlowski and Schmidt in around 1996, it looked at who looked to discuss or inform in around stress and the immune system. Acute stressors and physical activity have been shown to transiently enhance the immune responses. These stress effects on immunity seem to be mediated through endocrine factors, since hormones, neurotransmitters, and neuropeptides can interact with cellular components of the immune system. In works by Goebel and Mills in around 2000, who had looked at and investigated the effects of acute psychological stress and exhaustive physical activity on the expression of expression and density of adhesion molecules, L-selectin, LFA1, and ICAM1 on monocytes, granulocytes, granulocytes, and lymphocytes. Works by Segreger and colleagues, or collaborators. Segreger and team in 2000 highlighted in works in around chemokine receptors are present in are bases in acute allograft rejection, or AAR, and correlate with distribution of T cells. In works by Hancock and team in 2002, the team investigators informed that varied chemokine receptor expression, various chemokine receptor expression is evidence, or has been evidence, as part of cardiac allograft rejection. In works by Cockwell and team in 2002, who look at the importance and the role of chemokines and chemokine receptors in look at the importance of chemokines and chemokine receptors in acute allograft rejection, or AAR. In works by Larson and team in 2001. In 2001, the team inform of acute stress may signal an increase in at least some of the aspects of cell-mediated or T helper type 1 driven immune responses. In works by T helper cell type 1 driven immune responses. In works by Maddock and Parionte in 2001, they look at works that in around chronic stress, and where chronic stress appears to result in suppression of the immune response, whereas immune activation and suppression have been associated with acute stress. In works by Sternberg in 2001, who informed that physiological levels of glucocorticoids and immunomodulatory, rather than solely glucocorticoids, are immunomodulatory rather than solely immunosuppressive, causing a shift of cytokine production from a primarily pro-inflammatory to an anti-inflammatory response. Such responses of cytokine production can be characterized as T helper type 1 or T helper type 2, and roughly correspond to cellular or humoral immune responses. It works by Redwin and team in 2003. They hypothesized that activation of psychological stress and stress and responses increases autonomic activity and enhance immune function by inducing a significant increase in numbers of leukocytes at sites of inflammation. Redwin and team Redwin in works Redmond and team in works in 2003 also further informed that few human investigations have examined the effects of stress on leukocyte motility, and to date there have been little or no publications examining the psychological stress in regard to psychological stress and psychological stress and leukocyte chemotaxis. Chemotaxis did not correlate with did not or does not correlate with catecholamine levels, suggesting that another mechanism of activation may also exist, which increases chemotaxis after stress. In works by Yoon and team in 2004, we demonstrate that chemokine receptors play significant roles in the development of chronic rejection and may serve as potential therapeutic targets for cardiac allograft vasculopathy. In works by Xiufeng and team in 2005, they demonstrate the role, the unique role of chemokine molecules in the maintenance of cardiac allograft tolerance mediated by natural killer T cells in a murine model, in a murine or laboratory model. In works by Ulog and Callas in 2005, by Ulich and Callas in 2005, look at works where they inform that there is plurality of how neuroendocrinological factors can affect the influence of immunity and immune system mediated disease. There are consistent and convincing reports or works that link between stress and disease onset and progression. So prospectively, the perfect basis would be where recipients are stress free before transplantation, or where transplantation in around transplantation, i.e., for example, where on replacement, where for example, irrespective of being on forms of treatments or replacement therapies and prophylaxis, that throughout the time awaiting the time awaiting a transplant. And so this may well suppress those chemokine receptors that lead to acute allograft rejection or AAR episodes post-transplant. So now looking at a so looking at as part of this research poster presentation, a look at sort of the sort of basic biology, if you like, of chemokines and receptors. So chemokines and receptors in amongst literature, and primarily in works by Rossi and Zlotnick in around 2000, who in published works had informed of the sort of underlying biology of chemokines and receptors, thus where chemokines are involved in T helper type 1 cell and T helper type 2 cell development, are involved in angiogenesis and angiostasis, are involved in metastases, are involved in cell recruitment, are involved in inflammation, are involved in lymphoid organ development, and involved in lymphoid trafficking, and involved in wound healing. In part of this research poster presentation, I look at a sort of brief look at works looking at chemokines and receptors identified in human renal allograft rejection. So in works from Inston and Cockwell in around 2002, the investigators had in works looking at chemokine receptors identified in human renal allograft rejection, or AAR, chemokines such as rantes, interleukin 8, CXCL8, ENA 7/8, and CXCL5 have been findings expressed by endothelium and infiltrating cells and amongst infiltrating cells in acute allograft rejection, and where there's increased levels in urine amongst where acute allograft rejection is evident, and at the same time messenger RNA is increased in acute allograft rejection versus non-acute allograft rejection. And there's works by Patterson and team, sort of more retrospectively, in around 1994, and Mutabarak and team in around 1994, and Schmoder and team in around 1995 as well. In regard to chemokines, MCP1, CXCL2, IL8, CXCL8, and IL8, CXCL8, MCP1, CCL2, MIP, 1 beta, and CCL3. These are increased. Findings show that there's an increased expression, or there is evidence where there's a increased expression in tissue and levels in urine in acute allograft rejection, and increased levels of interleukin 8, for example, in acute allograft rejection, but all increased non-specifically in around acute allograft rejection. And there's works by Grandalino in around 1997, and works by Bud and team in 1997, and Sibbering and team in 1998. In regard to MCP1, CCL2, and MIP, MIP1 beta, and CCL3 MIP1 alpha, CCL3 rantes, and CCL5 findings have in amongst the evidence has shown that there's an increased expression of MCP1 and MIP1 beta by tubular cells with increasing grade of acute allograft rejection and increased expression of MIP and MIP1 alpha and rantes, but not related to the severity of acute allograft rejection, and works by Robertson retrospectively, Robertson and team, retrospectively in around 2000, in around 2000, had developed some underpinning works in regard to the findings sort of in around 2000, and then in works by in regard to chemokines IP10, CXCL10, rantes, and CCL5, MIP1 alpha, CCL3, MIP1 beta, CCL4, and lymphotactin XCL1. The findings have shown that there's that in acute allograft rejection, or AAR, the chemokines are all increased. And works by Segreger and team in around 2001 had evidenced some of the findings more retrospectively. Now, looking at chemokines IP10, CXCL10, rantes, CCL5, MIG, and CXCL9. The findings have informed that there are increased levels of acute allograft rejection in amongst biopsies, or where biopsies have been done, and infiltrating cells include CXCR3, where CXCR3 has been found positive, and there's works by Romaganani and team from 2001 who identified some of the findings in around 2001. In works in regard to chemokines CCR5, there's been findings in works where CCR5 expression is associated with mononuclear cell infiltrate in acute allograft rejection, and there's works by Etna and team in around 1998 that had identified some of the earlier findings in around CCR5. In around the chemokine CXCR4, findings have informed infiltrating cells in renal allograft rejection, and where CXCR4 is positive, and works again by Etna and team in 1998 had evidenced some of the findings in around CXCR4. And in regard to the chemokine CCR5, there has been findings in regard to co-localizations of CCR5 and T cells in acute allograft rejection, and works by Segreger and team in 1999 had informed some of the findings in around CCR5 in around 1999. And there's the chemokines DARC, which where findings have informed sort of an upregulation on endothelium in acute allograft rejection, and there is works There is works by a Segreger and team in 2000 who had identified some of the findings in around the chemokines DARC, and finally looking at chemokines CCR2, CCR5, and CXCR4, and in regard to CCR3 and CCR8. Our findings have informed there's an increase in that the chemokines increase in acute allograft rejection, although that the chemokines are increase in acute allograft rejection. However, CCR3 and CCR8 are absent in acute allograft rejection, and works by Segreger and team in 2001 had identified some of the findings in works in around CCR2, CCR5, CXCR4, and CCR3, CCR8 in around 2001. And so now finally, sort of looking at some of the before going into the sort of discussion, the discussion aspects, the discussion in around this research poster presentation, I look at some prospective investigations. So chemotaxis is a methodology to investigate transport or migration towards a concentration gradient of chemoattractant for leukocytes. And where leukocytes include neutrophils, monocytes, and lymphocytes, or other migratory cells. An upper chamber containing a suspension of cells is separated by a membrane from a lower chamber containing medium with chemoattractant. Chemotaxis of the cells of upper chamber into a lower chamber occurs over a predetermined duration. The cells in a bottom chamber may be counted directly when using homogeneous population of cells, or may be stained for defined markers when assaying a mixed population of cells and analyzed through flow cytometry. And looking at using a T cell culture system in isolation would be helpful. Using a T cell culture system in regard to chemotaxis. Chemotaxis would additionally be helpful. For in and around as follows. So number one, to observe chemotaxis for chemokine receptors, cellular adhesion molecules, and monoclonal antibodies, and using flow cytometry analysis to identify movement of particular and specific cells. And in regard, and secondly, measuring and looking at stress neuroendocrine levels pre and post transplant would be complementary investigations. 3, to monitoring sort of stress levels in pre-transplant duration is and can be of importance. Looking at number four, an investigation with chemotaxis assays could potentially observe catecholamines with repeated stress, and this would potentially allow to help identify which hormones, for example, influence chemokine receptor migration. Number 5, investigation that incorporates chemotaxis and monoclonal antibodies with neuroendocrine post-transplant would also be complementary investigations. Number 6, comparing the pre-transplant phase and observing chemokine receptors and neuroendocrine with monoclonal antibody therapy would be important, and with this, examining the effects of acute stress and exhaustive physical activity on chemokine receptor migration would be complementary. Number 7, studies or investigations with natural killer cell activity and cortisol with chemokine receptors in pre and post transplant phases may also be important. Number 8, conducting investigations that relate acute chronic stress cytokine activity and anxiety may show significant correlations between the immune system and neuroimmunobiology. And finally, 8. Measuring or looking at neuroendocrine function with hypothalamic adrenal axis, or IPTA, and chemokine receptor migration in pre and post transplanted recipients may be and could be important. And now finally looking at the discussion and then conclusions as part of this research poster presentation. So anxiety, anger, for example, anger and frustration are classed as acute stress and will cause an activated immune response, but not the same as positive stress. For example, motivational stress, positive stress or motivational stress, and physical activity propagates endogenous beta endorphin increase. Other neuroendocrine need to be better understood. Works by Sternberg as far as 2000 in around 2001 informed, for example, that glucocorticoids suppress cellular adhesion migration, macrophage activation and antigen presentation, T cell receptor expression, T lymphocyte activation, proliferation, differentiation, and including cytotoxicity, and B cell function, including antibody production. Chemokines are closely related to cytokines known as subcytokines, are produced by many cell types in response to infection or physical damage. Chemokines activate and direct cells to sites of tissue damage and regulate leukocyte movement into tissues. To date, professionals in around transplant, solid organ transplant specialties observe chemokines and IST in sole organ specialties, i.e., a nephrologist will sole-based investigations on renal pathophysiology and compare to investigations of other organ specialties. Previous investigations to understand acute allograft rejection and cytokines are commendable and results have proved progressive in works by Inston in 2000 in early 2000, around 2002, hypothesized that there is an alternative pathway that causes acute allograft rejection in recipients following anti-CD25 monoclonal antibody therapy. And works by Cockwell and team in 2002, around 2003, also hypothesized that in human transplantation there are alternative pathways to promote the development of a trafficking phenotype of T cells in patients or recipients that have received anti-CD25 monoclonal antibody therapy. In works at the same time where Cockwell in around 2002 2003 also utilized a T cell culture system to analyze the effects of anti-CD25 on the expression of chemokine receptor CXCR3 and other significant chemokine receptors on the surface of T cell. Currently, no research links neuroimmunobiology with neuroendocrinology in context of transplant immunobiology from a laboratory perspective. There is potential for prospective efforts in these areas, but would require multidisciplinary research efforts to acknowledge scope. Whilst there are complexities when regarding prolonging transplant allograft, whilst there are complexities when regarding prolonging transplant prognoses, transplant prognoses in published efforts or published works, in published efforts or research. one of the questions that has been put forward, would it not be sensible to acknowledge that stress can cause more of an activated immune response, and it is apparent that chronic stress and anxiety, for example, are linked and cause suppression of the immune system, whereas acute stress can potentially cause lymphocyte proliferation and initiation chemo and initiate chemokine receptor migration or trafficking to transplanted organ and instigate acute allograft rejection episode. However, at the same time, the link between stress and transplant rejection is uncertain. The postulation of using chemotaxis methodology would be admirable, opening point, and linking monoclonal antibodies with chemokine receptor migration and stress cellular adhesion molecules to identify how stress could potentially propagate acute allograft rejection. And it would be sensible to state that a stressed post-transplant population, that the stressed post-transplant population will have will have can have an increased cytokine production, and this could immunologically devastate allograft post-transplant allograft post-transplant around allograft post-transplant in around allograft sorry in around allograft post transplant, or cause another pathway for acute allograft rejection. And so finally, now looking at the conclusions as part of this research poster presentation from 2022. So more work is required linking IST monoclonal antibodies therapy, monoclonal antibody therapies, and cell surface targets and neuroimmunobiology with neuroendocrinology. The immune system has a suppressed action post-transplant, i.e., to reduce leukocyte proliferation, which would normally act against a stressor, for example, in regard to, or would normally act against a stressor, i.e., an organ transplant. So the aims around this works, or around this as part of this research poster presentation, were to establish and put forward an overview surrounding organ transplantation, neuroimmunobiology, and with neuroendocrinology in context of transplant immunobiology from a laboratory perspective. Whilst literature informs that chemokines surrounding transplantation provide a base, it is anticipated that this work could be used as a guide to prompt procedures prospectively across specialties. Using multidisciplinary research efforts should enable professionals to come together to achieve better understanding of transplant tolerance. Based on Selly's work in around 1936 and the general adaptation syndrome and stage 2, where the body interplays with defensive countermeasures against the stressor, it is postulated that chemokines appear to be integral players in this complex and dynamic process. A combination of neuroimmunobiology and neuroendocrinology in context of transplant immunobiology from a laboratory perspective to understand transplant immunobiology in context of stress, and Selly's works is important. That draws this audio podcast to a close. There will be a list of resources as part of this audio podcast and on social media. So and listeners out there are more than welcome to check out the literature around this works. That draws this audio podcast to a close. Then This audio podcast is part of a Phantom Edition. This audio podcast as part of a Vertex Roulet. This audio podcast as part of a Lobostics Machina. This audio podcast as part of a Storm Lobostics production.